Introduce our distinguished speaker, Diandra Mastrogiacomo. Diandra graduated with a Bachelor of Science in Biochemistry from the University of Tampa in 2021. Shortly thereafter, she advanced her studies by joining the Molecular Pharmacology and Physiology graduate program at the University of South Florida, where she has been making significant strides under the mentorship of Dr. Josh Scallon. Her research focuses on the molecular mechanisms controlling lymphatic valve development and is exploring how disruption in this process can cause human disease. Diandra's work has garnered several accolades, including the award for Best Oral Presentation at the Gordon Research Seminar in Lymphatics and the Molecular Pharmacology and Physiology Department Retreat. Additionally, she has been recognized with prestigious fellowships from the American Heart Association and the NIH. And the title of her presentation is Lymphatic Malformations with Activating KRAS Mutations, Impair Lymphatic Valve Development through MMPs. Please join me in welcoming Diandra.
Thank you so much for the introduction, Dr. Dellinger. So I'd like to start off by giving a quick overview of the lymphatic vasculature, which consists of lymph nodes all throughout the body, which are connected by collecting lymphatic vessels. The collecting lymphatic vessels are then connected to lymphatic capillaries. These lymphatic capillaries absorbs the excess interstitial fluid throughout your body. Once that fluid is absorbed, it becomes lymph fluid, and the lymph fluid is transferred through your collecting lymphatic vessels and lymph nodes, where it collects into your thoracic duct, which is the largest lymphatic vessel in your body. Then the lymph fluid is recycled back into your blood circulation through the subclavian vein. Collecting lymphatic vessels are characterized by the presence of a complete basement membrane, as well as a layer of smooth muscle cells, which contract to propel the lymph fluid forward. More importantly, these collecting lymphatic vessels have valves, and these valves prevent the backflow of fluid and allow for the unidirectionality of lymph flow. Lymphatic valves start to form around embryonic day 15 when oscillatory shear stress from lymph flow upregulates PROX1, FOXC2, and GATA2, which are all important transcription factors for valve development. Embryonic day 17, the valve leaflet starts to form when extracellular matrix proteins such as fibronectin, laminin alpha 5, or collagen type 4 are deposited. Embryonic day 18, the valve starts to mature. The end result is a bileaflet valve with 2 layers of lymphatic endothelial cells, also referred to as LECs, encapsulating an extracellular matrix core filled with those extracellular matrix proteins. Lymphatic malformations are a type of lymphatic disease with a dysfunctional lymphatic vasculature. They are usually characterized by the presence of vascular lesions filled with dilated lymphatic vessels. They are caused by post-zygotic mosaic mutations, and they're usually diagnosed in pediatric patients, but rarely in adulthood. The incidence is about 1 in 2,000 live births. Some other symptoms that these patients have include lymphedema, pericardial effusions, which is leakage of lymph fluid around the heart, chylosocieties, which is leakage of lymph fluid in the abdominal cavity, and here's an example of that in a mouse. And lastly, chylothorax, which is leakage of lymph fluid in the pleural cavity, which compresses the lungs, impairs respiration, and can lead to death if left untreated. And here's an example of chylothorax in a mouse. Activating mutations in the MAP kinase pathway have started to be identified in lymphatic malformation patients. So the MAP kinase pathway is shown here on the left, and the pathway starts with RAS bound to GDP in its inactive form. Once GTP replaces GDP, RAS is then activated and will activate RAF, and then RAF will phosphorylate and activate MEK, and MEK will phosphorylate and activate ERK. As you could see underlined in red, there are multiple mutations throughout this pathway, which causes excessive MAP kinase signaling that have been identified in these lymphatic malformation patients, such as mutations in the NRAS, KRAS, SOS1 and 2, CBL, and ARAF genes. Trametinib is a drug that is used to treat cancer patients, and it's a MEK inhibitor. Therefore, it has been repurposed to treat these patients to treat lymphatic malformation patients. But trametinib has toxic side effects. Therefore, we need to study this signaling pathway in more detail to identify downstream targets of ERK. So for our investigation, we use a FLT4 CREAR-T2 mice line, which is VEGFR3 and lymphatic specific. And we bred these mice with mice that have a stop codon flox in front of the KRAS G12D mutation. So then when we inject tamoxifen at P0 and P2, that stop codon is cut out and the activating KRAS G12D mutation is expressed in all LECs. We also use PROX1 GFP reporter mice to visualize the lymphatic vessels and valves. We analyze mice at P7, P14, or P21. When we were analyzing tamoxifen-injected P21 KRAS G12D mice, we frequently noticed chylothorax, and so this actually mimics what is seen in some patients with activating KRAS mutations. So to determine if chylothorax would cause an early death in these mice, we performed a survival analysis and found that the median survival time of these mice was 33 days. The thoracic cavity of each dead mouse contained chyle in 17 out of 17 mice. So in previous studies, chylothorax has been proposed to be caused by lymphatic valve loss via retrograde lymph flow into the chest. So we quantified valves per millimeter in the mesentery at P7, P14, and P21, and found a significant 62%, 73%, and 78% decrease in valves per millimeter, respectively. The total length of lymphatic vessels quantified did not significantly change at P7, P14, or P21 in the mesentery. So because lymphatic valves form during embryonic development in the mesentery before tamoxifen injection, these data indicate that the KRAS G12D mutation causes lymphatic valve regression. We also quantified valves per millimeter in the diaphragm at P7, P14, and P21, and found a significant 88%, 92%, and 93% decrease in valves per millimeter in the diaphragm, respectively. In the diaphragm at P7, there was a slight significant decrease in the total length of lymphatic vessels quantified, but there was no significant difference in the total length of vessels quantified in the diaphragm at P14 or P21 between the control and KRAS G12D groups. We also quantified valves per millimeter in the ear skin at P21 and found a significant 92% decrease, but there was no change in the total length of lymphatic vessels quantified. So because lymphatic vessels form postnatally in the diaphragm, and also because newborn pups are not even born with ears, the ears form completely postnatally, this data indicates that the KRAS G12D mutation prevents postnatal valve formation. So this actually recapitulates data that Dr. Michael Dellinger and his lab have published using a PROX1 CREAR-T2 line. So to conclude what we've learned so far, existing valves are regressing over time in the mesentery of KRAS G12D mice. The KRAS G12D mutation prevents postnatal lymphatic valve formation in the diaphragm and ear skin. And the KRAS G12D mice do not live past 8 weeks old due to chylothorax. So we want to know, what is the mechanism underlying valve loss in lymphatic malformations? So we turn to the literature, and this paper was published in the New England Journal of Medicine in 2018. They identified activating KRAS mutations in patients with brain arteriovenous malformations. So for their investigation, they performed immunostaining with human umbilical vein endothelial cells, which were cultured, and they performed immunostaining for V-cadherin, which is the cell-to-cell junction protein, as well as phosphorylated ERK. And they treated these cells with lentivirus to express the activating KRAS G12D mutation. And the lentivirus that they used had LymphaC-GFP, which binds to F-actin, as you can see here in green. So all the cells shown in green are cells that are infected and expressing that activating KRAS G12D mutation. So the mutated cells, as you can see here, do not have V-cadherin localized at the cell membrane like we see here in the control cells. And then when they treated these cells with a MEK inhibitor, this rescued that phenotype, and V cadherin was once again localized back at the cell membrane. This is another paper that was published in Nature Medicine in 2019, and they identified an activating ARAF mutation in a patient with a central conducting lymphatic anomaly. Here is immunostaining of cultured LECs expressing that activating ARAF mutation, and they also performed immunostaining for V cadherin. These cells expressing the activating ARAF mutation do not have V cadherin localized at the cell membrane, like we see here in the control cells. But again, when they treated with trametinib, a MEK inhibitor, this rescued that phenotype, and V cadherin was once again localized back at the cell membrane. So overall, what we see from these two papers indicates that hyperactive MAP kinase signaling causes either internalization, degradation, or downregulation of V cadherin. So we wanted to investigate this, and we performed immunostaining on cultured LECs expressing, we treated them with lentivirus to express either a control vector or the activating KRAS G12D mutation. And we also performed immunostaining for V cadherin. And as you can see, V cadherin was not localized at the cell membrane and the LECs expressing KRAS G12D, but PROX1 expression was maintained, as you can see in green. So why is this important? Well, our lab published this paper in 2019, in which we identified V cadherin as being essential for lymphatic valve formation and maintenance. We used a PROX1 CREAR-T2 line, and we knocked out V cadherin and quantified total valve number and found a significant 90% decrease in total valve number in the mesentery at P14 when V cadherin was absent. The valves that were remaining had an abnormal morphology, as you can see here in panel P. Overall, this paper discovered that the absence of V cadherin causes the regression of lymphatic valves as well as prevents postnatal valve formation, therefore phenocopying what we see in the KRAS G12D mice. So all in all, this paper concluded that V cadherin regulates mechanotransduction signaling in the lymphatic vasculature because it is required for beta-catenin and AKT signaling. And then beta-catenin and AKT signaling are required for PROX1 and FOXY2 expression, which are the important transcription factors for valve development. So when V cadherin is absent, beta-catenin and AKT signaling do not occur, PROX1 and FOXY2 are not expressed, and thus valves are unable to form. So we hypothesized that hyperactive KRAS signaling was inhibiting localization of V cadherin at the cell membrane, thus inhibiting mechanotransduction signaling and causing the loss of lymphatic valves. So to investigate this, we generated mice which have the monomeric M-scarlet red fluorescent protein inserted into the end of exon 12 right before the stop codon of the gene encoding V cadherin, thus creating a good reporter mouse for V cadherin that is easy to stain with using a red fluorescent protein antibody, also referred to as an RFP antibody. So when we stain with RFP in these M-scarlet mice, we have very beautiful visualization of the veins, arteries, and most importantly, the lymphatic vessels and valves. So we used these M-scarlet mice and bred them with the KRAS G12D mice, performed home immunostaining with an RFP antibody and confocal imaging, and we saw the obvious presence of V cadherin at the cell membrane and the mesentery at P14, and in the diaphragm and ear skin at P21 and the KRAS G12D mice. To further confirm these results, we performed immunostaining using an antibody specific for V cadherin and the regular KRAS G12D mice with no M-scarlet. And again, we confirmed the presence of V cadherin at the cell membrane and the mesentery at P14, and in the diaphragm and ear skin at P21 and the KRAS G12D mice. So our preliminary data indicates that the loss of V cadherin at the cell membrane does not occur in vivo in the KRAS G12D mice. So our hypothesis about V cadherin is insufficient to explain valve loss. So we're trying to figure out what's causing valve loss in lymphatic malformations. So here's RNA sequencing data generated by our collaborator, Dr. Michael Dellinger in his lab, from cultured LECs expressing the activating KRAS G12D mutation. Upon looking it over, we noticed that a number of MMPs were upregulated, as well as components of the plasminogen activator pathway. The plasminogen activator pathway starts with plasminogen, synthesized and secreted by the liver, and it circulates through your blood. And then TPA and UPA serine protease enzymes are secreted by endothelial cells, which are then, these enzymes then cleave and activate plasminogen into plasmin. UPA activation requires the binding of its receptor UPAR. And then plasminogen activator inhibitors PI1 and 2 inhibit both the TPA and UPA enzymes. So UPA is mainly involved in processes such as cell migration, invasion, angiogenesis, or tissue remodeling. And TPA is mainly involved in the process of fibrinolysis, which is the dissolving of blood clots. So then plasmin will go on to degrade fibrin clots into fibrin degradation products, and then plasmin will cleave and activate pro-MMPs into MMPs, and then plasmin and both MMPs degrade extracellular matrix proteins. So we realized that these MMPs that were upregulated from the RNA sequencing data, they degrade the same extracellular matrix proteins found in the core of the valve. And these proteins are important for the development and maintenance of lymphatic valves. So we hypothesized that hyperactive KRAS signaling was upregulating MMPs and components of the plasminogen activator pathway, such as TPA, UPA, and UPAR. And this was leading to excessive activation of MMPs and causing degradation of the extracellular matrix core of lymphatic valves, and thus resulting in lymphatic valve loss. So to confirm the RNA sequencing data, we performed quantitative real-time PCR with cultured LECs treated with lentivirus to express either a control vector, or the activating KRAS G12D mutation, or wild-type KRAS as another control. We confirmed the upregulation of TPA, UPA, and UPAR in the LECs expressing KRAS G12D. SERPIN-E1 is the plasminogen activator inhibitor that is expressed in LECs, but the expression of SERPIN-E1 did not change in the LECs expressing KRAS G12D, therefore indicating that the SERPIN-E1 expression does not affect this upregulation of the plasminogen activator pathway that we see here. We also confirmed the upregulation of MMP1, 2, 9, 10, and 14 in the LACs expressing KRAS G12D. We also performed Western blot to confirm the upregulation of UPAR, the receptor for UPA, and MMP14 on the protein level in LECs expressing KRAS G12D. So as you can see, the LECs expressing wild-type KRAS had a slight upregulation of UPAR and MMP14 when compared to the LECs expressing the control vector, but the LECs expressing KRAS G12D had a higher expression of UPAR and MMP14 even when compared to the cells expressing wild-type KRAS. To assess the expression of secreted MMPs, we collected conditioned media that was incubated with serum-free media supplemented with plasminogen for 18 hours. And so after collecting the conditioned media, we used that as our samples for Western blot and performed Western blot for MMP1 and MMP10. And so in this LECs expressing KRAS G12D, there was an upregulation of active MMP1 and MMP10, because as you can see, the band is of a lower molecular weight, therefore indicating that these MMPs are activated. The LECs expressing wild-type KRAS had a slight upregulation of MMP1 and 10, but only in the latent version, and only when compared to the LECs expressing the control vector. So if TPA and UPA enzymes are present in the LECs expressing KRAS G12D, then they would cleave and activate plasminogen into plasmin, and that would result in the MMP1 and MMP10 being activated in only the LECs expressing KRAS G12D. Therefore, these results strongly support our hypothesis. We also performed gelatin zymography using the conditioned media in the same methods that we described in the previous slide. So gelatin zymography is a SDS page enzyme assay in which we use a gel made from gelatin, and we use conditioned media as the samples, and we perform electrophoresis to separate the proteins. And as the proteins travel down the gel, any gelatinases present will degrade the gelatin. So then we perform Kumasi blue staining, and areas of degradation will appear as clear bands against a darkly stained background. So in the LECs expressing KRAS G12D, MMP2 and MMP9 were present, but not present in the control LECs and the LECs expressing wild-type KRAS. So to further investigate the presence of TPA and UPA enzymes, as well as MMP activity, we have been collaborating with the Davis Lab here at USF, and they've been performing experiments for us using their 3D culture system in which LECs form interconnected tubes that are suspended in a collagen matrix gel. So they are treating these LECs that are expressing and activating KRAS G12D mutation with plasminogen. And when they add plasminogen, the collagen gel contracts and degrades into a small ball, as you can see here. And this process is accelerated with increasing concentrations of plasminogen. These results were quantified by determining if the gel was not contracted at all, partially contracted, or fully contracted. And so at higher concentrations of plasminogen, the gel was fully contracted, but at lower concentrations of plasminogen, the gel was partially contracted. They also treated these these LECs expressing the activating KRAS G12V mutation with aproteinin, a plasmin inhibitor, and TIMP1, an MMP inhibitor. And so when they treated these LECs with aproteinin or TIMP1, this rescued that phenotype and prevented the contraction and degradation of the collagen gel into that small ball. And these results were quantified after 3 days. Overall, these data indicate that that plasminogen is sufficient for MMP activation and collagen degradation. So to assess in vivo expression of the plasminogen activator pathway, we harvested the mesentery at P21, performed whole mount immunostaining, and then confocal imaging. We stained for UPAR, the receptor for UPA, and it appears that UPAR is upregulated in the mesentery at P21 and the KRAS G12D mice. We also performed immunostaining for laminin alpha 5, one of the extracellular matrix proteins found in the core of the valve, but it's also found in the basement membrane. There appeared to be less laminin alpha 5 present in the valves and the basement membrane of lymphatic vessels in the KRAS G12D mice. Lastly, we stained for fibronectin, which is another extracellular matrix protein found in the core of the valve. There appeared to be less fibronectin present in the remaining valves of the KRAS G12D mice. We also harvested the diaphragm at P21 and performed immunostaining again for UPAR, and found that UPAR appeared to be upregulated in the diaphragm at P21 in the KRAS G12D mice. We also performed immunostaining for laminin alpha 5 in the ear skin at P21, and there appeared to be less laminin alpha 5 present in the basement membrane of lymphatic vessels in the KRAS G12D mice. So because the 3D culture experiments that Dr. Davis and his lab performed indicated that plasminogen was sufficient for MMP activation and collagen degradation, we wanted to see if knocking out plasminogen would rescue lymphatic valve loss in the KRAS G12D mice. So we bred global plasminogen knockout mice with the KRAS G12D mice and quantified valves per millimeter. We found no significant difference in valves per millimeter between the KRAS G12D mice and the KRAS G12D mice with plasminogen knocked out. We also harvested the diaphragm at P21 and found no significant difference in valves per millimeter between the KRAS G12D mice and the global plasminogen knockout mice with expressing KRAS G12D. And lastly, we also harvested the ear skin at P21 and again found no significant difference between the KRAS G12D mice and the KRAS G12D mice with plasminogen knocked out. So overall, these data indicate that plasminogen is not required for MMP activation and lymphatic valve loss. because it did not rescue valve loss. So because other serine protease enzymes can activate MMPs other than plasmin, we wanted to see if directly inhibiting MMPs would rescue lymphatic valve loss. So we treated the KRAS G12D mice with alomostat, a pan-MMP inhibitor, which inhibits all of the upregulated MMPs except for MMP10. So we treated the mice with alomostat from P0 to P6 and harvested the diaphragm at P7. There was no significant difference in valves per millimeter between the vehicle-treated and alomostat-treated KRAS G12D mice. So we thought maybe it takes longer for the drug to work, and so we harvested tissues also at P14 after treating mice with alomostat from P0 to P13. There was still no significant difference in valves per millimeter between the vehicle-treated and the alomostat-treated KRAS G12D mice. Therefore, these data indicate that alomostat treatment does not rescue lymphatic valve loss in the diaphragm of KRAS G12D mice. But we also harvested the mesentery at P7 after treating with alomostat every day from P0 to P6. And when we quantified valves per millimeter, we found that there was a significant increase in valves per millimeter in the alomostat-treated KRAS G12D mice when compared to the vehicle-treated KRAS G12D mice. More importantly, there was no significant difference between the vehicle-treated control mice and the alomostat-treated KRAS G12D mice, therefore indicating that alomostat treatment fully rescues lymphatic valve loss in the mesentery at P7 of the KRAS G12D mice. We also harvested the mesentery at P14 after treating with alomostat from P0 to P13. Although there was still a significant decrease of valves per millimeter in the alomostat-treated KRAS G12D mice when compared to vehicle-treated control mice, there was still a significant 2.3-fold increase in valves per millimeter in the alomostat-treated KRAS G12D mice when compared to the vehicle-treated control mice. So these data indicate that alomostat treatment can rescue lymphatic valve loss in the mesentery at P14. So to conclude what we've learned from this project, KRAS G12D mice do not live past 8 weeks old due to chylothorax. Lymphatic valves regress in the mesentery and prevents valve formation in the diaphragm and ear skin in KRAS G12D mice. V coherent is still present in KRAS G12D mice and cannot account for valve loss. The plasminogen activator pathway and MMPs are upregulated in cultured LECs expressing the KRAS G12D mutation. UPAR appears to be upregulated in the KRAS G12D mice. There appears to be less fibronectin and laminin alpha 5 in these mice. Knocking out plasminogen does not rescue lymphatic valve loss in the KRAS G12D mice, but treating with alomostat, the MMP inhibitor, can rescue the loss of lymphatic valves only in the mesentery of these mice. Overall, this project will allow for the identification of new combinatorial treatments for life-threatening lymphatic malformations by pharmacologically inhibiting the downstream targets that directly cause valve loss. I would like to thank the organizers of this seminar for giving me the opportunity to give this talk. I would, of course, like to thank Dr. Scowen, my PI, and our entire lab. I'd like to thank Dr. Young and her entire lab. I'd like to thank my committee. I'd like to thank Dr. Michael Dellinger and his lab at UT Southwestern. I'd like to thank our collaborators, Dr. Davis, Dr. Hong, and Dr. Sorokin. I'd like to thank our funding sources from the DoD, NIH, and AHA. And I'd like to thank everyone so much for listening. I would love to take questions now.
Great presentation, Diandra. So it's
Thank you.
open for questions. So if you have a question, you can either use the react button to like raise your hand, or you can just unmute yourself and ask your question. All right, we'll start with Sarah.
Hi, really wonderful work, and it's been so exciting to see you present this at different conferences along the way. I was really wondering if you followed out your alomostat treated mice out and looked at chylothorax in them and the survival.
Thank you so much for your comment. And then to answer your question, yes, so the alomostat treated mice have never gotten chylothorax. but unfortunately, alomestat is just really toxic for them. So they don't live up to P21 usually.
Thank you.
Mhm.
Can I ask another question? Sorry.
Of course.
Do you have any other thoughts, since that seemed to help them, in terms of restoring the valve function in the mesentery? But it sounds like it was toxic from, I guess, another perspective for them. Do you have another thought about any other inhibitors or any other drugs you might try, kind of in a continuation of this work?
So in general, like I've read that the inhibitors for MMPs are becoming more specific over time, which is, great news. But in addition to that, there was a paper that treated a patient with I think it was an arteriovenous malformation with murimistat, which is another MMP inhibitor, and the patient experienced a resolved symptoms. So it actually helped them. So that was promising.
Well, thank you. Really, really exciting work.
Thank you so much.
I have a question, Diandra. Phil King here in Michigan. I would be more formal and raise my hand in the chat group, but I don't know how to do it.
It's okay.
So I was wondering if the increase in that you have observed in vivo, or Michael's lab had observed in vivo, of the MMPs can be blocked by MEK inhibitors. Is this actually a consequence of dysregulated MAP kinase signaling? Is at the heart of my question.
Yes, so I'm pretty sure Josh could comment on this. We did look over RNA sequencing data from LACs treated with trametinib, and I'm pretty sure that trametinib treatment did decrease that upregulation of MMPs that we saw. But we looked at it a while ago, so I'm not entirely sure. So I don't know if Josh remembers, he could tell me if he does or not.
Yes, I think you have it correctly. We have also a single cell, or not single cell. We have RNA-seq results where Mike's lab treated them with trametinib, and it looks like the plasminogen activator pathway, when it goes up, it also goes back down with trametinib. So yes.
The disconnect, therefore, is that if in our hands, if we use long-term MEK inhibitors in an adult mouse that has formed valves, that the valves become disrupted. They lose function, they regress. This is a consequence of long-term administration of those inhibitors. Whereas if this MMP pathway were downstream of MEK, you'd expect the opposite effect, right?
Yes.
See my meaning?
You would expect more valves, is what you're saying.
Yes.
Yeah. So we do see that in the mesentery. I don't know if you saw the data when Diandra presented it really quickly, but if you look at control mice that are treated with alomestat, we do see an increase in valves. So.
Right.
Yeah.
Let me think about that.
Okay. Thank you for the question.
Yes, thank you.
Well, since nobody else is asking a question, just a quick comment. Very happy to see that you can't confirm the VE cadherin changes, because to me, disruption of junctional integrity is always like a go-to that many labs engage in as an explanation, molecular explanation for their findings, whereas in fact that could just be downstream of a number of other more immediate consequences. So we have never, in the absence of negative regulators of RAS in mice, seen any disruption of junctional integrity, at least not in a mouse that was not half dead. In our initial paper, more than 13 years ago now, we did report upon some VE cadherin maldistribution in the absence of RAS1. But the truth is that was probably in half-dead animals or dead animals, and the maldistribution was probably consequent to impending cell death. The truth is that whenever we've tried to look for changes in junctional integrity in the face of dysregulated RAS-MAP kinase signaling, we've never really seen them. And I know that a lot of other groups working in the blood vasculature have different opinions. But certainly in the lymphatic vasculature, we're not confident that that happens.
I mean, I think it's a great point that you brought up. So we know Diandra didn't mention it in the presentation, but we also know that our mice don't develop chylothorax, which is what you would expect if you had problems with junctional integrity. So it's a little bit different than knocking out V cadherin, where we do have chylothorax. But we didn't see any issues with V cadherin expression in these mice, and the main way also that you get chylothorax, as Diandra mentioned, is that at least that the lymphatic field seems to agree on, is that you lose valves, and the loss of valves leads to chylothorax. It's not really a loss of junctional integrity.
Yeah.
Yeah. Thank you.
Yes, thank you.
All right. Well, if there are no other questions, I just want to thank you once again, Diandra, for an excellent presentation. And I'm just really so happy with the past two presentations by young investigators and seeing the future of the field of complex lymphatic anomalies. So thank you once again.
Well, thank you for giving me the
Mhm.
opportunity. Appreciate it.